Myself, Mohammad Muzaffarur Rahman, Assistant Professor, School of Pharmacy, Nalla Narasimha Reddy Educational Society. In the previous video, I have said I'll be dealing with microbiology subject, isn't it? So, from the series of lectures on the microbiology, today is our second lecture. In the previous video, we have seen what microorganisms are, how are they characterized, what are their applications. We have also have had a brief introduction on the history of microbiology, the main scientists who are contributing or who has contributed a lot in the field of microbiology. In the characteristics of microorganisms, we have seen their size and also we have seen if they are multicellular or unicellular, aerobic, anaerobic, and we have also mentioned a point about prokaryotes and eukaryotes. So in today's lecture, we'll be seeing what prokaryotes and eukaryotes are. And we'll also see what are their structural organization within them, how are they different from prokaryotes and eukaryotes. So from today's lecture, the main objective of today's lecture is to identify what prokaryotes and eukaryotes are and differentiate between the prokaryotes and eukaryotes and find what are the similarities between prokaryotes and eukaryotes. Both the organisms are have cells, right? So they have certain cell organelles as well. So we'll see what are those common cell organelles present. Okay, and next we'll see the components of a cell, and we'll see functions of various cell organelles along with the components of a cell. And after completing the presentation, we'll see a small video of the or internal organization of a cell. Okay, beginning with what prokaryotes and eukaryotes are. So prokaryotes, you can break the word into two, that is pro and karyotes. Pro means before. For example, let us take primitive humans. Considering Homo sapiens or humans, primitive humans are the early man, isn't it? Similarly, prokaryotes means pro before, karyot means nut or kernel. When do we call nut? Suppose we have consider a walnut. Okay, there is some material, edible material inside it which we eat, and above it is the covering, the outer covering, that is called nut or kernel in botany. So that covering is called as nucleus or nuclear membrane in the microbiology. So you can see here prokaryotes means before nucleus. Before nucleus, why do we say that? Because the early primitive species of microorganisms doesn't have covered nuclear membrane. So we'll see in different characteristics of prokaryotes. First thing, these are unicellular organisms that lack membrane bound nucleus. We have just discussed it before nucleus, right? That means before nuclear membrane. So here, these are unicellular organisms which lacks membrane bound nucleus. And next, all cell organelles including DNA and RNA are present in the cytoplasm. As I have just said, there is no nucleus present. Therefore, the genetic material such as DNA or RNA are present within cytoplasm. And these are very smaller in size, that is 0.1 to 10 micrometer. And you can see all bacteria are prokaryotes. Moving on to the eukaryotes, what does U mean? U means true. True nucleus. That means it has a proper nuclear membrane. We'll see the characteristics of eukaryotes now. These are unicellular or multicellular. Unicellular examples can be amoeba, paramecium. These are unicellular, right? Whereas multicellular, you can consider human beings or plants or animals, anything, right? So these are unicellular microorganisms or organisms, unicellular or multicellular organisms that possess membrane-bound nucleus. That means true nucleus. And therefore, the genetic material are present within the nucleus since it has a nuclear membrane and possess various organelles that helps in cell function. We'll see what are those special cell organelles as well in the coming slides. And these are larger than prokaryotes, that is the size is between 10 to 100 micrometer. And the examples of eukaryotes can be all plants, animals, fungi, are eukaryotes. They possess eukaryotic cells. So what are the difference between the prokaryotes and eukaryotes? You can see the structure here. This is a prokaryotic cell and this is the eukaryotic cell. This is a plant cell. Uh, yeah, this is of animal cell and this is a prokaryote. Okay, a bacterial cell. So these are unicellular, small size. These are the differences between it. Here. The prokaryotes are unicellular, while eukaryotes are multicellular. Smaller in size, the size range 0.1 to 10 micrometer. Here there are larger in size range from 100 to 10 to 100 micrometer. Membrane bound organelles are absent. You can see everything is present randomly within the cytoplasm, whereas here membrane bound organelles are present. There is a nucleus separately here. Okay, you can see here nucleus separately, and every organelles are separated right within the cytoplasm. They are given a specific place for that. And prokaryotes produce by asexual reproduction, while eukaryotes produce either by asexual or sexual reproduction. We'll discuss in detail the reproduction process in brief when we study bacteria, fungi separately. Okay, and these prokaryotes contain DNA as genetic material, and this DNA is circular DNA. Circular DNA, which means they are also called as plasmids. Okay, and here we have linear DNA. In the video, we'll see how eukaryote DNA is the double helical structure that you have studied in your intermediate. And basic proteins are associated with DNA, the genetic material. The basic proteins are associated. While in eukaryotes, histone proteins are associated with DNA. Suppose there are four histone bodies, two like this and the two other like this. The DNA material will be coiled around the histone proteins. Okay, and it has 70S ribosomes, whereas eukaryotes has 80S ribosomes, and contains cytoskeleton is absent, whereas cytoskeleton is present. All these points will be covered when we see the video of the internal organization of a cell. Moving on, what are the cell organelles that are common? So first thing, all these are the characteristics of eukaryotes, and this one is the characteristics of prokaryotes. This is a Venn diagram that highlights the common organelles present. First is flagella. What is flagella? Flagella are hair-like projections which helps in movement of a cell. In prokaryotes, it is flagella, whereas in eukaryotes, it can be flagella or cilia. Flagella in eukaryotes, let us consider a human being itself. The flagella is present only to the sperm cell. It helps in motility of a sperm cell. Whereas cilia is present in many cells, which helps in transport of mucus from the cell lining. Okay, that will be a best example to describe the motility of cells. And the other one is plasma membrane, or you can call cell membrane. And cell division is different in prokaryotes as well as in eukaryotes, but there is cell division. For that reason it is considered as a common one. And then cytoplasm present in both prokaryotes and eukaryotes, and ribosomes are present both in prokaryotes and eukaryotes, and also chromosomes. Okay, and now we'll move on to the next one, the components of a cell. So you can see that this is a basic structure of an animal cell, right? And here is the nucleus, the pore. The pores that you can see are the nuclear pores, and this is the nucleolus. We'll study in brief the structure of nucleus and its function. And you can see this is the endoplasmic reticulum, and even here you can see there are certain dots on endoplasmic reticulum. This is rough endoplasmic reticulum, while this one is smooth endoplasmic reticulum. And you can see the mitochondria here, cytoplasm, ribosomes separately, lysosomes, vacuoles, and cell membrane. So we'll begin with the studying the structure of cell membrane. Okay, cell membrane. As you know, cell membrane is a bilipid model or sandwich model, which means one bread is above, the other is below, and there is something here that we can consider as a jam between two breads, right? So the upper side is lipophilic and the lower side is lipophilic, whereas the middle part, which is the tail part, is hydrophilic in nature. You can see the structure here. So this is the yellow part is the hydrophobic above and below. That means the sandwich model. and the middle part is hydrophilic part. And you can see the blue color structures, they are the receptors, or all receptors are proteins, right? So all these receptors are responsible to entrap the drug molecules or any other molecules such as carbohydrates or lipids. You can see the attachment of a carbohydrate here to the protein or the receptor. You can see the glycoprotein or globular protein and glycolipid attached, and cholesterol that is passing. Cholesterol is highly lipophilic, isn't it? So it is easy, it can easily be permeable to the cell membrane. And these are the different proteins through which transport of ions take place. Okay, let us see the functions of a cell membrane. It is first is selectively permeable. As I've said, it helps in transport of ions between the cell, external cells, and keeps cell organelles together. As you know, cell membrane covers the whole entire cell. As a result, the organelles present within the cell membrane are intact. And recognition of external substances, it helps in understanding, because of the receptors present, it can understand if the glycoprotein is attaching or carbohydrate is attaching, because the receptor for glycoprotein is different and the receptor for carbohydrate is different and the receptor for lipid is different. Thereby it can understand what compound is being attached to the cell membrane, and site for various chemical reactions. And the attachment of a drug or any compound to the receptor itself causes certain reaction. As you know, enzyme substrate complex. When a substrate come and attaches to the enzyme, there will be a conformation in the enzyme, isn't it? So there is a chemical reaction going on. So all the receptor sites are site of recognition of various chemical reactions. Moving on to the structure of mitochondria. Mitochondria is called as the powerhouse of the cell, whether it be plant cell, animals, or any organism. They are responsible to generate amount of ATP, large amount of ATP that is required for our body to produce or to work energy, to work, as we need energy that is provided by ATP. Well, let us see the structure of a mitochondria. It is a double membrane structure. Okay, there is an intermembrane space between two membranes, and the composition of intermembrane space is that of cytoplasm present in plants, or cytoplasm present in animal cells. It is similar to the that of normal cell cytoplasm and the intermembrane space cytoplasm. And the outer membrane is permeable to certain ions and small molecules. And inner membrane, in inner membrane we have studied various cycles that are going on, such as Krebs cycle or beta oxidation. All these occur in the internal membrane of the mitochondria, and folded into multiple cristae. You can see the way the outer way are called the cristae and the inner one are called the matrix. So here the outer one is a cristae and the inner one is the matrix. Let us see the function. It is essential for aerobic respiration. Aerobic respiration, that is by electron transport chain, then we go for oxidative phosphorylation where ATP are generated, right? So aerobic respiration that occurs in presence of oxygen, and energy production via oxidative phosphorylation, as I've just said, and it is involved in various metabolic pathways such as beta oxidation and Krebs cycle. Beta oxidation is the oxidation of the lipids. Next, moving on to the another cell organelle, that is endoplasmic reticulum. So you can see this one. This is the nucleus or nuclear membrane in which nucleolus will be present. To this attached is the endoplasmic reticulum. These are the projections that you can see is the endoplasmic reticulum, and there are certain space called as cisternal space. Okay, and cisternae, the outer projections, and the ribosomes are present on the surface of the endoplasmic reticulum. If ribosomes are present on the endoplasmic reticulum, they are called as rough endoplasmic reticulum. If the ribosomes are absent on the endoplasmic reticulum, they are called smooth endoplasmic reticulum. You can see here these are the rough endoplasmic reticulum, right? Whereas these are the smooth endoplasmic reticulum. Let us see the function. These ribosomes are responsible to synthesize proteins. Okay, and it helps in lipid synthesis, as well as it produces enzymes and microtubule production, which maintains the integrity of the cell. Now we'll study what ribosomes actually are. Ribosomes are of two types. It can be 70S or 80S. In prokaryotic cell, it is 70S. That means 30 subunit and 50 subunit. 30 is a smaller subunit, 50 is the larger subunit. So uh, let us consider this a smaller subunit. To this attached is the larger subunit. This is 30S plus 50S. This is 70S. Similar to that, eukaryotic 40S and 60S. You can see the structure here, simplified ribosome here. When modeled in a uh, software, you can see in this way. This is the smaller subunit, this is the larger subunit, and molecular level. If we see each amino acid in that way, it will be in this way: smaller subunit and the larger subunit. Let us see how it is organized. So as I've said, smaller subunit is in the bottom, the larger subunit is in the top of above the smaller subunit. This is a 50S and 30S in case of prokaryotes, whereas in eukaryotic ribosomes, 60S is the above one, that is the larger part, whereas the 40S is a smaller subunit. Okay, both are attached in this way. Let us see what are the functions. Functions of the ribosomes are to synthesize proteins. First, it sequences amino acid one above the other. As you can see the image here, this mRNA gets amino acid, okay, and transports here, keeps the amino acid here, and these amino acids goes on attaches here, okay. As a result, the chain keeps on increasing. Same way, this pRNA attaches here, the amino acid comes here and goes up. The other one comes here, attaches here, that amino acid gets attached here and again goes up. Similarly, another tRNA comes here, this amino acid goes and attaches here, this will increase the chain. So sequence of amino acid is called a protein, right? Similarly, here all the amino acids are sequenced one above the other because of the ribosomes, and as a result it produces proteins. And these proteins or the sequence of amino acids get converted to a tertiary structure called as a quaternary structure, and we can also call as post translation changes that we'll study in later on. Okay, that is called translation process, in where the uh, proteins are formed such that it forms a complex structure, that is called post translation changes. And this is the translation that it happens, that is the synthesis of proteins from the sequence of amino acids, or you can say it is a production of proteins from the genetic material or from RNA. Moving on to the Golgi apparatus. These are smooth, uh, as we have seen the structure of smooth endoplasmic reticulum, they are similar to that. You can see here the smaller parts are called vesicles. These vesicles, how are they formed, we'll see in the video. So you can see the structure: incoming transport vesicle. There is an empty space called lumen. The outer surfaces are called cisternae, as that of endoplasmic reticulum. And newly forming, these are the small projections that are newly forming the vesicles. It first entraps certain things which are to be eliminated, and then slowly comes out in this way and form a new vesicle. So you can see the newly formed vesicle is a secretory vesicle. We'll see the function of it. It modifies proteins and lipids, okay, and major sulfur carbohydrate synthesis, we'll see in the video, and process materials to be removed from the cell. As I've just said, unwanted materials will be taken up by the Golgi apparatus, and that will be given out in form of vesicles. That vesicles will be consumed by lysosomes and it will be removed. We'll see that in video how it actually happens. And then it make and secrete mucus and packages products into vesicles for transport. As I've said, it removes unwanted particles or unwanted substances within the vesicle and takes away. So it is also responsible for the transport of materials. So let us see your lysosomes. What are lysosomes? Lysosomes are small molecules. You can see the structure here. It has certain enzymes, especially um, lyso hydrolyzing enzymes. Lysosome contain hydrolyzing enzymes. These hydrolyzing enzymes are responsible for the, lysis of many other things. So we'll see how it happens. First, bacteria. Let us consider bacteria as a foreign organism. It is a foreign organism, right? It takes the bacteria through the receptor, and this forms in this way phagosome. Phagosome secretes or gets absorbed into the phagosome and cause the lysis of the cell. You can see this red color part, which is a bigger one, and here when it comes, it breaks into smaller. That means the lysozyme and the lysosome has released certain hydrolytic enzymes that has broken this bacterial cell. As a result, soluble debris it is removed by exocytosis. Let us see the functions now. Contains hydrolyzing enzymes and responsible for the hydrolysis of proteins, fats, etc. Called autophagy, that is self-destruction when it consumes certain bacteria. Fuses with vacuoles to digest food. Right? It is fusing with vacuoles. As I've said earlier, the vacuoles are the vesicles. It combines with the lysosome, and thereby it gets degraded, and it helps digest unwanted particles, as you have seen this example, and helps white blood cells to destroy bacteria. Okay, we'll see that in the video. Then the last but not the least is the nucleus. Nucleus is the most important part in the cell. It contains genetic material. Okay, you can see the structure of a nucleus. This is the endoplasmic reticulum. So this one is a nucleoplasm. The one here, nucleoplasm. The which means the cytoplasm present within the nucleus is called nucleoplasm. And the second one is nucleolus. This nucleolus contains genetic material. And third one is nuclear envelope. The outer membrane of the nucleus is called nuclear envelope. And nuclear pore. There are certain pores on the nucleus which are responsible for the release of ribosomes from the nucleolus. We'll see that in the video. So this is the image of the nucleolus. You can see the nucleolus covered by nuclear membrane, and it contains the genetic material such as DNA or RNA. Let us see the function. It contains genetic material, right? Contains instruction to make proteins. How does it get instructions? Since it has genetic material, that genetic material gets converted to single stranded DNA, then it produces RNA. That RNA goes to the ribosomes, right? Those ribosomes produce proteins. So it has instructions to produce different types of proteins. And nucleolus makes RNA and ribosomes that I have said earlier. Now we shall proceed to the video where you can understand very clearly how the entire process takes and how each and every cell organism is responsible for certain activities. Let's now consider this is a human cell or animal cell. Organs are specialized parts of cell, right? We'll see now, beginning with the nucleus, which is the control center of the cell, and it contains a genetic material, that is DNA. As I have said earlier, it contains double-stranded DNA, right? And we'll see how it is going to act. This DNA forms chromatin. If we see from afar, it looks like a thread-like structure, and that are called chromatin. Okay, and that is the nuclear membrane, and this chromatin moves together, condenses to form chromosomes, right? Okay, the nucleus also contains nucleolus, I've discussed earlier, which produces many ribosomes. These ribosomes are coming out of the nuclear membrane and attaches to the endoplasmic reticulum and forms proteins or synthesizes proteins. These ribosomes float in the cytoplasm, and you can see the cytoplasm around the endoplasmic reticulum. And okay, and this is the rough endoplasmic reticulum, as I have said earlier. The ribosomes attach to that to form rough endoplasmic reticulum, and here you can see smooth endoplasmic reticulum, as ribosomes are absent on that. And these are the within the endoplasmic reticulum, the proteins that are being synthesized are given out in form of vesicles, proteins or other materials. These vesicles gets into the Golgi apparatus, as you can see, Golgi apparatus or Golgi body, where these proteins further forms a complex structure, that is a tertiary structure. These are a sequence, right? It forms a complex structure and forms a tertiary. And this tertiary structure, to that lipid or carbohydrates can be attached. Next is the lysosome. You can see how lysosomes are. They contain hydrolytic enzymes, right? They first collect certain unwanted materials and then dissolve in it due to presence of hydrolytic enzymes and forms a cellular debris which removes by exocytosis. And these are mitochondria, which is a powerhouse of the cell that produces lots of ATP required for the work, ATP for energy and also for respiration, cellular respiration. So during this process, ATP will be produced, especially by oxidative phosphorylation, right? So that ATP will be consumed by cells. And for the cell to maintain its integrity, it needs a cytoskeleton, right? These cytoskeleton are made from microfilaments. These are a thread-like structure present within that, or we can also call as microtubules, okay, which are hollow from inside. As discussed earlier, I have said certain flagella or thread-like structures which helps in movement are present within the cell. Could be a flagella. It could be a flagella in case of prokaryotes, whereas in eukaryotes could be flagella as well as cilia. In flagella, it helps in movement of the entire cell, right? In human beings, cells contain cilia that are, which are finger-like projections that helps in movement of nucleus. You can now see this video now. This is the cilia that is moving forth and back to and fro to move the mucus above during cough. And this is a cell of bacteria which is a prokaryotes and it has flagella that helps in moving forward. And this is a sperm cell which contains flagella. The only cell in human being to have flagella is the sperm cell. So this is it with today's class. The next class will be studying the structure of bacteria, its internal organization, and how they can be identified, what are different types of bacteria, classification of the bacteria. Okay, and thank you for watching the video.